So this is representing mitosis. We start off with 2 chromosomes, one being from the dad and one being from the mom, or 2. So we both have 2 chromosomes here from each, which makes it diploid. And what diploid is what I already said, 2 chromosomes, one from the mom and one from the dad. Why we have different lengths is this is chromosome 1 and this is chromosome 2, and 1 is the longest, and then it goes from the next longest and so on. What these are is the centromere, which connects the 2 genes together. These beads representing genes, but in the human body we have way more than what is represented here. We call these chromosomes homologous because they are not from the same person, as in mom and dad, but they do have the same length. So these 2, one from the mom, one from the dad, same length, also with these 2 chromosomes as well. So the goal of mitosis is to make an exact copy of the nucleus. So as the cells get bigger, they copy its DNA. So as I said before, as the cell gets bigger, the DNA is copied, which is all happening in G1, which is part of interphase. So right now we have all the chromosomes, which we are going to duplicate into the same exact, but one more of each. All right, so after they copy themselves, we enter S phase, which makes the exact copy of each one. As you can tell, there's the same amount of genes for each. So this is called sister chromatids, as there is one chromosome and another of the exact form formed together, making those sister chromatids, which is normally how we see chromosomes in this stage look like. All right, so before we get into prophase, we have to enter G2 phase, which is the growth phase. So each chromosome and sister chromatid will form into many more in that one nucleus. But now we are seeing prophase, is where we actually see the DNA to combine, and that we see that little X shape we're normally used to seeing. After this, we hit metaphase. So we now see in metaphase that the chromatids line up, and from each centromere, which is what binds the 2 together, we make spindles to the side, and each of these go out into the nucleus and they connect like this. And usually these spindles will go all the way outside the nucleus, but since I'm on a piece of paper, it looks like this. It's kind of hard to see since these beads are the same color, but these spindles will actually line everything up in the middle, which makes the metaphase plate. After this, we go into anaphase, which these spindles will shorten, and this is what that looks like. So this is representing anaphase. It's kind of hard for me to show the spindle shortening, but basically what happened here was from that previous image, when they're all aligned, they will actually form, and the spindles will get shorter and shorter and break off, and forming these 2 separate, eventually nucleus. So as I said, these will form into 2 different nuclei, which is now into the telophase. So now we'll have 2 nuclei. So what I'm going to do is I'm going to draw a circle on each side to represent its splitting, which is end of mitosis.